Hello class, welcome back. Today we will be discussing the same chapter that we had started last time, that's chapter 7, respiration in plants. In our previous class we have already discussed about the general idea of respiration, like what is respiration, what are the conditions required for respiration, what are the types of respiration in detail, right? Today we will be discussing about different experiments related to respiration. Okay, there are certain criteria or conditions which are required for respiration. Whether those conditions are met or not, in order to discuss about that, we will be discussing few experiments related to it. Okay, now let's carry on with the experiment. Okay, so let's carry on with the experiment. First experiment is to prove that oxygen is used up in respiration. Okay, if you go through the general meaning or definition of respiration, what is it? Respiration is a process in which glucose is broken down in the presence of oxygen to release heat and energy. So, in order to prove whether oxygen is really used up during respiration or not, this particular experiment is carried out. Okay. Now, before carrying out any experiment, for experiment there are two types of setups. One setup is called experiment setup, another one is called control setup. Now both of it is identical, that means they are similar. All the conditions are same in both the case, except in case of control setup, whatever you are trying to study in experiment, that's missing. Okay. Now similarly in this experiment also you can see setup A and setup B. Setup A is your experiment setup. Setup B is your control setup. Okay. Now, if in both two conical flasks are used and both the conical flask has a bed made up of wet cotton wool. Why wet? In order to provide water to the seed, because for seed to germinate you need water. Okay. That's why cotton wool are both the cotton wool are wet. Okay. Both the condition is same, you can see here. Now second thing is, now setup A has a germinating seed, whereas setup B has a dead beans. Why is setup A having a germinating seed? Because setup A is experimental setup and only germinating seed would be carrying out the respiration. So in case of setup A, so you want to study the respiration process is whether using oxygen or not. So for that you need to have a germinating seed, because germinating seed would be carrying out the respiration process. So you have germinating seed beans in case of setup A. Okay, but in setup B it's a control, so you have dead beans. Compare dead beans to non-living, any non-living thing. Will non-living thing carry out respiration? No. Similarly your dead beans will also be not carrying out respiration. So in case of the setup B, whatever oxygen was present inside the conical flask, it will remain as it is. But in case of setup A, your oxygen would be used up by the germinating beans in order to carry out the respiration. Okay. Now in both the flask you can see that small test tube is placed, right? You can see a small test tube is placed. Now it consists of soda lime. Soda lime is a mixture of sodium hydroxide and select lime. Okay. Why is it used? It is used so that if any, because we know that during respiration carbon dioxide is produced. So if any carbon dioxide is produced, it is absorbed by the soda lime. Okay. Now, and you have two delivery tube coming out of setup A and setup B, right? You have delivery tube. Now that delivery tube is connected to the container. You can see a container which is filled with the water. Okay, this is your experimental setup. You keep this for a few days. After a few days, what would you observe is, delivery tube which is connected to setup A, there would be a rise in the level of water. You can see here in the experiment also, there is a rise in the level of water. But delivery tube which is connected to B, there you cannot see, even if there is a rise, there will be a slight rise in the level of the water, but as compared to A, it's you cannot, it's very less. Okay. Now why does that happen is because in case of setup A, you have a germinating seeds, right? So germinating seeds would be utilizing the oxygen which was present inside the flask, okay, in order to carry out the respiration. So vacuum would be created in this area, okay. Now because of that vacuum pressure, water from this container will be raising up in order to fill that vacuum space, okay, in order to fill that empty space. That's why there would be a rise in the level of water in case of setup A. But in case of setup B, since no respiration is going to take place because seeds that are placed are dead, okay, so whatever the level of oxygen is there, it will not be utilized, right? So no empty vacuum created, so no rise in the level of the water, okay. Now other thing, in order to prove that oxygen is used up or not, what can you do is, you can remove this cork, okay, and then bring a burning flame in on the mouth of both the flasks. In case of flask A, what would you observe is immediately your flame would go off, okay, but in case of flask B, it would light up for a while, okay. Why does that happen? Because in case of flask A, no oxygen is present, okay, because it has been used up by the germinating seeds, and oxygen is essential, oxygen supports burning, right, but there is no oxygen, so that's why your flame goes off immediately. But in case of flask B, you had oxygen and oxygen supports burning, that's why your flame was glowing for a while, okay. So this experiment proves that oxygen is used up in respiration, okay. I hope you understood this particular experiment, because this application based question, most of the time from this chapter they ask you from this experiments only. This is the major topic of this chapter. All the experiments you have to go through, all the experiments that's present in this chapter. Why? And while carrying out the experiment, you should be able to reason out why is soda lime present, why are they using wet cotton instead of dry cotton, okay. And in setup B, why are they using antiseptic? Why are they using antiseptic? Because dead beans are present, okay, they would be decayed, okay, by the bacteria. So in order to prevent that, because antiseptic, what do? They kill off the bacteria, right? So in order to prevent the decay of the dead beans, okay, that's why they are using the antiseptic, okay. So you should be able to reason out why. So that's all about this experiment. Now you have one more other experiments. Now another experiment is to prove that carbon dioxide is produced during respiration. Like I told you, during respiration carbon dioxide is one of the byproduct, right? So for that also, like I told you, in all the experiment you have control and a experimental setup. The one round bottom flask, setup A is your experimental setup, setup B is your control, because in case of setup B you have a boiled beans, okay. Boiled beans is not going to carry out any kind of respiration, right? So your setup B is your control and setup A is your experimental setup. In both the flask you have placed wet cotton wool, okay, and then in flask A you have placed a soaked bean because that would undergo germination, and on the flask you have placed boiled bean. No respiration is going to take place, okay. Now you have a lime water, okay. After a few days, what would you do is you remove the cork, okay. For separately for A and B, you take a test tube having lime water. Now A, you pour out the water extract from A into the lime water. In case of A, your lime water would be turning milky, okay. Now that proves that carbon dioxide was produced during respiration. Now in case of B, no respiration takes place, no carbon dioxide is produced, so your lime water would not turn milky, it would remain as it is, okay. So how would you prove that carbon dioxide is produced during respiration? You are using lime water, because lime water when reacts with the carbon dioxide turns water milky, okay. So if lime water turns milky, that means carbon dioxide was present, okay. So that's all about this experiment. The another experiment fulfilling the same condition, that is carbon dioxide is produced during respiration. Okay. Now another experiment to prove the same condition, that is carbon dioxide is produced during germination. Okay, you have 4 flasks set up. Flask A has potassium hydroxide solution, then B has lime water, C has germinating seeds, and D has again lime water. Now you pump in the air from the outside, okay, atmosphere. Now flask A has a potassium hydroxide solution. What does it do? It makes sure that if there is any carbon dioxide present in air, then it would be dissolved by it is dissolved in potassium hydroxide solution, and whatever the air is coming out of flask A would be devoid of carbon dioxide. Okay, to make sure that air does not contain carbon dioxide, in flask B you have lime water. Okay, now air goes on through that flask. Now lime water in the presence of carbon dioxide, what happens? What did I tell you? It turns milky. But in this case the water was clear, that means your air doesn't contain any carbon dioxide. Okay, now you have a germinating seeds there. That flask B is connected through a delivery tube to flask C. It has germinating seeds. Okay, now oxygen is supplied, so germination would take place, or respiration is going to take place. Now during respiration carbon dioxide is produced. Okay, and delivery tube C would carry the whatever if gas if present in the flask C to flask D. Now flask D has a lime water. Okay, now this lime water would turn out to be milky. Now till when this delivery flask A was connected to flask B, the lime water did not turn milky because there was no carbon dioxide. The same air has been transferred to flask C, but when this flask C is being connected to flask D, the lime water in flask D is turning out to be milky. Now that proves that during respiration carbon dioxide is produced. That's why the lime water in flask D is turning out to be milky. Okay. Another experiment to prove that green plants produce carbon dioxide during respiration. It's similar to that we have studied earlier, but instead of beans we are using plant itself. Okay. So you have a plant which is covered with a bell jar, and you have 2 delivery tubes connecting to the flask. Okay. First what would you do? The air would be sucked inside. In that delivery tube we have a soda lime, because soda lime would be absorbing the carbon dioxide if at all present in air. Okay. Now the air is the delivery tube coming carrying the air from outside is connected to flask A. It has lime water, so lime water would turn out to be clear. Okay, because soda lime has absorbed carbon dioxide already. Okay, now that delivery tube is connected to your bell jar, inside which you have a green plant. Now green plant would undergo respiration, it would produce carbon dioxide, right? So there is another delivery tube coming out of the bell jar. Okay, and it is connected to your another flask B, and it also has a lime water. Now what would you observe? This is the lime water in flask B would turn out to be milky. Now what does that prove? That proves that carbon dioxide was produced here, that is carbon dioxide was produced by green plants. Okay, that's why the lime water in flask B is turning out to be milky. So what does this prove? This proves that carbon dioxide is produced by green plant during respiration. Okay, these 3 experiments that we have been doing are similar. Okay, just that in this experiment they have used green plants instead of your germinating seeds. Okay. Now another experiment is to prove that heat is evolved during respiration. Okay, you take 2 thermos flasks, one is your experimental setup, another is your control. Both the flasks, in one experimental setup flask A you are putting a germinating seed, in B you are putting a dead beans with antiseptic. Okay, and you have inserted a thermometer in both the flasks. Why are you using thermometer? Because thermometer, if there is any heat produced, thermometer would indicate that. Okay, now you keep this setup for a day. Okay, after a day, what would you see is, in experiment A, where you had placed a germinating seed, there was a raise in the level of mercury in a thermometer. But in case of B, since no experiment is being respiration is being carried out, because dead beans are present, so no, slight or no rise in the thermometer can be, level of the mercury in a thermometer can be seen. So this proves that during respiration your heat is evolved. Okay. Now another experiment, like I told you, there are two types of respiration, one is aerobic and another is anaerobic. Aerobic means glucose is broken down to produce energy, or glucose is broken down in the presence of oxygen to produce heat and energy. And anaerobic respiration, here oxygen is, in the scarcity of the oxygen, this particular respiration is carried out. Okay. Now for plants, if anaerobic respiration is carried out, ethyl alcohol is produced. Okay. And partial breakdown of glucose only takes place. That's why only 2 molecules of ATP is produced during anaerobic respiration. Now you have two setup, one experimental setup and one experimental setup. One setup, you have one experimental setup. What would you do is, you take a test tube, you place a bean in it, germinating seeds, and then fill it with a mercury, and then in a flask, okay, what do, you would, instead of water, you are putting a mercury. Okay. Now then invert the test tube and then place it in the conical flask. Okay. Now what would you observe? Why are you using mercury? In order to maintain an anaerobic environment, because water, then it would be broken down into hydrogen and oxygen would be provided, right? That's why in order to avoid that, you are using a mercury here. Okay. You keep this setup for few days, 2-3 days. After 2-3 days, what would you see is, vacuum is created here. Okay. Now why does that happen? Because these seeds, they undergo respiration, anaerobic respiration, carbon dioxide is produced, gases rise up. Okay. That's why you have a clear vacuum is created here. To prove that whether that particular vacuum was out of carbon dioxide or not, what you do is, you would put a potassium hydroxide in it. Potassium hydroxide, what would it do? It would dissolve the carbon dioxide, and again the mercury level would be filled up till the top of this test tube. Again it would be filled with the mercury, because whatever the carbon dioxide was produced during the experiment, it has been dissolved by potassium hydroxide. Okay. Now that's all about the different experiment. You have to go through all these experiment. Okay. It's very much important. From this chapter, most of the time the questions are asked from this experiment only. Okay. So that's all about the different experiment related to respiration in plant. So we have already discussed about different experiments related to respiration. Now two important phenomena that takes place in plant is your photosynthesis and respiration, right? So I guess you will be able to differentiate between what is photosynthesis and what is respiration. Photosynthesis, what is it? It's a process in which green plants prepare the food. Okay. And what is here, the glucose is produced. And in case of respiration, what is it? It's a process in which energy is liberated. So here glucose is broken down. So for your photosynthesis is an anabolic process, whereas your respiration is a catabolic process. Now in case of photosynthesis, you need chlorophyll, sunlight, but that's not essential in case of respiration. Okay. Your photosynthesis takes place only during daytime, but respiration takes place both during day as well as night. Okay. Now I guess you will be able to yourself be able to analyze the difference between both this phenomenon, that is respiration and your respiration and photosynthesis. So that's all about the chapter. You please go through different experiments, that's very much important from this chapter, difference between respiration and photosynthesis. Okay. And next I will follow it up with the question and answer related to this chapter. Thank you.